Robert, like that though.
They've seen him a little bit here and there. And so what happens on Green Jeans Parent Day, it's a little bit different from the other Green Jeans. I mean from the other courses like Fun with DNA and World of Enzymes, because basically you get the idea where you come in and you get the demonstrations of all the labs that they did. Now in Green Jeans though, a lot of the labs that they do are kind of based on the labs that they've done before because they're now applying it to what they need to do. And so rather than have you watch, like for example, how to do a restriction analysis all over again, we're just going to kind of discuss the aspects of the course so you've got an idea of what happened this week. And so we're going to go in and they're going to do some oral, like little oral presentations with some props, and there's also an animation we're going to throw in there, and we're going to talk about what happened during the week. And then after that, they're actually going to get to show you part of the experiment that you've never seen before, and that's how to purify the protein, the glowing protein that they've been working and striving for all week. And so yesterday, we actually purified the protein that we were looking for it, and then overnight, we grew some colored fluorescent proteins, and so they're going to purify some really exciting colored protein for you guys today. And so now, so Green Jeans, I'll tell you about how the course runs and what it's all about once we get into the other classrooms. I've got some cool pictures to show you. And while I'm here, though, I'll tell you a little bit about what happens after this. So now, once your child sequence organisms, trying to just kind of look at sequences and compare them, and a group of students in Manhattan stumbled upon a sequence of DNA that was different from all the other types of DNA, all the other types of creatures that lived with it. And it turns out that what they uncovered, they actually uncovered an undiscovered species of cockroach in Manhattan.
We all need more cockroaches.
Yes, we need to know about what types of cockroaches they have in Manhattan. I'm not very surprised that they found a new species in Manhattan, but anyway. And so, or it was, but there was a group of snails that they thought this one species of snails went extinct. And this one scientist, he was actually a graduate student, said, "That's weird, why would this one snail go extinct when all the other species around it didn't? What happened there?" So he decided to get in his kayak with some friends, and they kayaked up the river, and they went to the area where they thought these snails were, and they found the extinct species alive. There it was. And so, yeah, so this is something that's kind of exciting on the horizon of DNA research and things like that. And so that's DNA barcoding. Then after that, you go into the high school, and so what you have in common with other people. And then we go into plant genomics, where we kind of look at different plants, and we look how they've been modified. There's a lot of genetically modified organisms, plants that we eat every day or that we consume every day. And also, we have a few new things. There's a course called Silencing Genomes, which is exciting. It actually works with a worm called C. elegans, a little microscopic worm, and which probably, if you guys took Fun with DNA, you might have come in contact with that little worm, saw it under a microscope. And so basically what they do with these worms is they found out that if you introduce a certain type of strand of DNA into the worm, you can silence a gene. So you can turn a gene off. Okay. We already did our welcome to Green Jeans, so I want to tell you a little bit about the course. And so the whole idea of the course was to try to purify protein. And so we wanted to see if we could create a protein, and now proteins are created basically by your cells. So your DNA is the genetic cookbook for all the proteins that your body makes. We make millions of different proteins. And so you make these different proteins that have different functions. So we have proteins that make us run. We have proteins that give us color, things like that. And so for this, we wanted to make a protein that's easy to see. And so technically, we can make any protein we want with this method, but we wanted to be able to see it this week. So we chose a protein that's made by this creature right here. So that's our, who can tell me what that guy is? That guy is jellyfish. And this jellyfish is called Acoria victoria, and it has this neat trait of being able to glow in the dark. And so it glows in the dark because it actually makes a protein called green fluorescent protein. And green fluorescent protein, basically when scientists discovered that they could take that protein out of cells, and then when they discovered that they can take the instructions to make it, they kind of took off and did many different things. And so one of the things that they've done is basically, these are more of that jellyfish right there, is the first thing they did was they were able to take that gene and put it into bacteria. So what you see right here is a plate of bacteria growing that are glowing. And so now the protein by itself, though, doesn't glow under, like it doesn't glow in the dark, a jellyfish does, but they have other genes that kind of help them to do that. And so therefore, if you take the protein out, you need to expose it to ultraviolet light. And so you need to shine some UV rays on it in order for it to glow. And so these are basically, these little bacteria cells are being exposed to UV light. And so then they started to basically take that gene and they started to put it into organisms. And so now this is like one of the most recent types of organisms that they were able to put it in. Look at something, what are those little things? Yes, they're rhesus monkeys, exactly. And so, yep, they made glowing rhesus monkeys as a baby reduce like these them. And so now you might think, well, what's the point of that? And so, well, yeah, some of that, but in other words, they had to perfect this. They had to get this down to the science because now they have new uses for it. And so they started to use it for what we call reporting. And what it does is they've gotten the GFP to be inserted into cancer cells. And so when you're looking for these cancer cells, basically they'll glow under UV light. And this is something that they're perfecting. This is my laboratory, the Cold Spring Harbor Laboratory, does a lot of work with cancer, and this is one of the things that they're working with today, is using GFP to kind of target cancer cells. And what it's enabled them to do is to do better surgeries, so that they get more of it, and they get like all of the cancer, so there's less chance of it growing back and things like that. And so that's one reason why they were working with GFP so closely. And then another scientist, he actually won the Nobel Prize for this, where he figured out that if you take the gene for glowing protein, you can fix it a little bit, you can change the little recipe just enough to change its color. And so right here, this is also a plate of bacteria. These bacteria, when you basically put them on the plate, they're so tiny that they grow in place. And so this is different color. Each of these little colors are made by a different type of bacteria with basically a mutated form of GFP. And so they've got a slightly changed version of the GFP. So they made RFP, there's purple glowing protein, there's blue, we didn't have blue. But you know, green and brown, and they have all these colors. And so now his research, though, he went beyond just bacteria as what he's studying, and he's using this for neuroscience. And so he's using this to study brain cells. And so when he's got all these brain cells, he said, well, how can you tell which branches are which? And so basically now, he gives each brain cell its own color. And so it's sort of see if we could make any protein. And so in the end, we're going to go through all the steps. We had some success. We had some, well, failure, not on their part, but there was a part where, well, nothing decided to grow. And then, or very little decided to grow. And then also we had great success at the end to the protein purification table right in the corner right there. So go ahead.
First, we took our gray pipette and scraped a colony off an agar plate that looks like this. And then we took the entire tube and colony and we put it in another tube with Luria broth and left it overnight in an incubator at room temperature, or body temperature.
Transfer 1 milliliter of the overnight culture into the microcentrifuge, and then
What the bug buster does, it breaks apart the bacteria so all of the proteins that are inside can flow out, and that's what we want, is just the GFP protein purified. After that, we would centrifuge that after adding the bug buster, so all of the bacterial debris would collect at the bottom, and basically what we have on the top of the supernane is a solution of just the proteins from the bacteria. So then we would have to separate the other proteins that the bacteria contains just from the GFP. We took first a sample of the supernane and put it into a separate tube. We added something that's called binding buffer. And basically what the binding buffer would do is it would attract the GFP to small hydrophilic beads, and that would allow the GFP to float down to the bottom of the tube when centrifuged and separate from the rest of the proteins. So once the beads were added and the GFP connected with the beads, the tube was centrifuged and the remaining proteins collected at the top while the GFP was at the bottom of the tube. We added later 800 microliters of wash buffer to the bead, which basically would help us get rid of the excess proteins that were on top, anything but the GFP. And finally, we added 200 microliters of elution buffer, which would separate the beads from the GFP. So once you got rid of the beads from that tube, all you had, the liquid there, was pure GFP. And we tested it in the end by putting it over a UV light, and that's how we see if we had a purified GFP.
Excellent, very good. And so what they're going to do, and your presentations were fantastic. Oh, I want to tell you about one other experiment that we did, and I threw this in there because it's just fun, and you can see an example of it. Well, right here. So they did some bacterial painting. And so when they got a chance, and I gave them 2 chances, the first try sometimes, it runs and you gotta get used to it. And so the second, we have some lovely results. And you'll get a chance to kind of see some of the bacterial art that they produced and look at it under a UV light. It looks really fantastic. And for kicks, do this. It's glowing green.
So this is the protein she made?
Yeah. So she's got some glowing protein in there.
And it's alive?
Well, yeah. I'm wondering if the genes are in there and it's not bacteria and it's yeast, because yeast can use the plasmid and glow green also. Then that means they ate your bacteria that had the right genes and they're glowing green. That'd be really funny if that's true. But you definitely have glowing protein there. That's the color that we would be looking for.
Excellent.
So I thought you'd get a kick out of that before you left.
See, I made the protein and it glows.
But tell me, why is she one blue and one green?
Because this is, it's different colors. It's the same thing though.
Oh,
Where is it blue?
the right side.
This one?
In my camcorder.
Yeah, it's supposed to be this color.
Okay, so that looks good.
This is green fluorescent, like the one that everyone has, and this is the purple one that I made on ice. Protein or something that I
I'm not
made.
sure if something ate the yeast or it is the yeast itself.
I made it. And
With the protein then? What is this?
no, this is a different one. Like just my name with it's protein, but it's like I made a picture with it.
Okay, but it's not fluorescent, doesn't glow in the dark?
No, it does. It's glowing.
Oh yeah.